Hello, everyone, and welcome to this week's bytesize talk. With us today is Evangelos Karatsas, and he is going to talk about the newest release of NF-core protein families. He is currently working at EMBL in Cambridge, but he's talking to us from his home place, right?
Yeah,
So it's all yours now.
Thank you, Francisca. Let me share. There we go. So, thank you for the introduction. I'm currently part of the Magnify team in EMBL EBI, Rob Finn's group that's situated in the UK. But yes, as you said, I'm currently in my old lab in Alexander Fleming back in Greece, doing my long secondment here, praying for some more sun before I leave. So yes, actually, this is the first release of protein families. And to let you in on the subject, I will briefly talk about the background, what we're doing here. Let's say everything starts from samples. You can have a sample from human gut, or then you can have another sample from spider, amphibian samples, or a lake, or God forbid, everything together. An environmental sample that has everything, proteins from everything inside. So yeah, let's say that we skip a big part of the analysis process and we end up with our proteins, which is the input point, the entry point for protein families. But of course, we don't have the 3D structure of it. But first, the analysis gives us a sequence of a protein. Let's say that this is a human protein. And then you might have another homologue of this protein and from another human that's very, very similar, but not exactly the same. So these proteins can easily be aligned. And then let's say that you have a mammalian protein that's also related, and then you add some gaps here, and that's also aligned to the original proteins. You can have multiple of these, put them inside, let's say, this alignment that we have here, and even have some similar proteins that are coming from bacteria that are not so closely related, but still they are part of the family. And this might not have exactly the same function, but some rare cases they can be, for example, let's say one bacterial protein can withstand extra heat. So this makes it extra special for this protein family. So we can have multiple of these different proteins in the same family. This is what we call a multiple sequence alignment, or short MSA. And for each position, we can get the probability of an amino acid to be there. And if we calculate that, we can create what we say the hidden Markov model, the HMM of the family. For example, for this multiple sequence alignment, along with many, many other proteins that we're not showing here, we can have specific amino acids with a specific probability in every position. The bigger the letter of the model, the bigger the probability for the amino acid to be there. So after we create this HMM, we can use it to recruit, for example, more similar proteins to the family. But if we have, like, let's say, a very trashy, let's say that's very random sequences put together, this will make the generation of the HMM, well, the generated HMM will be very sensitive, so it will be recruiting garbage in our families. And on the other hand, if all our proteins are very, very strictly homologous, let's say, very, very similar to each other, this will make the HMM very specific. So it will only recruit certain orthologs in the family. So let's see what the protein families pipeline does and why, step by step. First, yes, it generates protein families. And we're doing that to understand, for example, the evolutionary changes between different proteins among different species, and see what drives these evolutions, what these alterations in the amino acids imply. Then another thing that the pipeline does is that it expands the sequence space of already existing families. We have an update mechanism, so we can take already existing families give a new protein FASTA input to the pipeline, and this can check if these proteins can extend and expand the already existing families. This is a means to capture the sequence diversity, add extra proteins of different organisms in the same family, and study functional differences of these proteins. And finally, the ultimate goal is to identify novel and families that have uncharacterized proteins inside them. This will let us explore the dark matter of proteins, which is a very huge chunk of the protein space, and hopefully uncover new functions in the end. But protein families is not directly doing that, this uncovery of new functions, the discovery, but it is a task that can be handed over after the generation of the families. It can be handed over to downstream NF core pipelines, such as the protein annotator, which is a baby pipeline. Let's say it's in its infancy right now, but the goal is to be able to feed FASTA files with proteins to this pipeline, and then this will try to annotate the sequences that we give. So anything that remains unannotated might be really novel. So this is a means to discover novel enzymes and drug targets that will enable therapeutic applications and biotechnology innovations in the long run. So now we'll guide you through the workflow itself, the pipeline. The first thing, well, the input for the pipeline, as we already discussed, is a FASTA file with input proteins. Then the first stage is the initial clustering, which we're currently doing with the MMSEX suite. We create a database in the MMSEX format. Then we let the user choose the clustering algorithm of choice, either the MMSEX cluster, which is more sensitive, but it's extremely slow for large datasets. And then the link last method, which is extremely fast, but might give smaller cluster points. So yeah, it's up to the user to decide, and depending on the size of the input FASTA file. And then this results into the clustering file with 2 columns, which we will decide on which clusters we will focus on depending on thresholds such as minimum cluster membership. Moving on, the second sub-workflow, let's say, of the pipeline is the family generation sub-workflow. After we have filtered our initial clusters, we allow the user to choose between 2 aligners, 2 alignment algorithms. The first one is the MAFT software, and the second one is the FAMSA align software. Then optionally, the user can clip the generated alignments. This means that if there are gaps either on the sides or wherever in the sequence, this can be removed above certain thresholds of gappiness. And then we are left with what we call a seed multiple sequence alignment, which is used as input in HMM build from the Hammershoot to create the HMM of the family. Then we use this HMM to refine, let's say, the families via HMM search, where we use the input FASTA again, and through the model of the family, we recruit extra sequences in the family. And then we align these sequences with HMM align to the original model, and we output what we say the full alignment of the family. Then the third sub-workflow is completely optional. It consists of two different parts. The first one checks for family redundancy, meaning we remove whole families that are redundant or duplicates. And the other half removes in-family redundance, which means that we remove very similar sequences from inside the same family. So for the first one, we are doing an HMM search of the family representative sequences against all generated family models until now. And if we find very high identical hits, meaning we're using a stricter threshold this time to match our proteins to the models. If we find similarities, we discard those families currently that are smaller in size, those with less members. Then we're using a stricter version of the original clustering for each family. to remove the intra-family redundancy, the sequence redundancy. By doing this strict clustering, we keep only the sequence representatives, and then we are left with fewer sequences that still describe different huge range of organisms. And then we realign these sequences that remain to update the full multiple sequence alignment file. There is a complementary mode that's called update existing families. If the user provides extra HMM files and MSA files from previous runs with the family models and alignments, the FASTA file that we provide to the pipeline is checked with an HMM search again against the existing families. And if there are hits, we append these sequences to the existing families. We create again strict clustering to remove redundancy, and then realign, clip again, and update the HMMs of the families. And for any sequences that didn't have any match, it goes back. This is what these arrows mean. The original flow goes back, and for the remaining sequences, it just follows the original method of generating protein families. Currently, the multi-UC outputs are not very rich because none of the modules that we're using currently provide automated multi-UC reports. So we provide our own. One for the initial clustering, which shows the size distribution of the originally created clusters from MSX, how many clusters have size of 1, and how many clusters here, 29 clusters, have cluster size of 3. This is what it means. And then the final output, let's say the final metadata output for the whole family, this is per sample, and this is per run. We have the output family IDs, their size, the representative sequence length, its ID, and the corresponding sequence. So after we finalized the first version of the pipeline, we did some benchmarks to test if there are any bottlenecks, see how fast it goes. So we did a medium one, let's say, with UNIREF 50 that has 68 million proteins, which finally concluded into 15 non-redundant families. This might seem very small, but because we use identity threshold of 50 in the pipeline, and this is already UNIREF clustered at 50% identity, this is kind of expected. And large example where we used proteins from UNIREF 90. This is UNIREF clustered at 90% identity. This has almost 200 million proteins that resulted in almost 5,000 non-redundant families. So with a first glance, we can see some outliers here on the execution. For example, for the locally created chunk clusters module, this seems quite lengthy in comparison to the other modules. So what we did here is we swapped the SecIO library, the original one that was used to read the proteins, with PyFastX, and we managed to quarter the duration of the pipeline of the specific module. This has already been done in the dev branch of the pipeline. Following, we can see that sometimes the FAMSA align module takes way too long. And the plan is to try another module, Trimal, and put a hard cap of the maximum number of, before we call the aligner, we trim the sequences that we feed to the aligner with a hard cap, so we do not waste all these resources. Then HMM search, this also takes, sometimes it takes longer than expected. We will try using Nail, which is a kind of new software that promises the sensitivity of HMM search, but at the speed of MSX search, which is very, very nice. This does not have yet the bioconda environment, so it will be a little more difficult, but we will do it. create the environment and then the NF-core module and put it in the pipeline. And then the filter recruited local module, again, seems to be taking longer sometimes. And we want to try a combination of PyHammer and PyFastX instead of the currently manual parsing that we do, along with Python's SecIO. And finally, for the extract family reps, we have various different methods in mind. One we want to try is to extract the family representatives earlier, because we can do that from HMM search results, or try doing it with Trimal instead of the slow by Python SecIO that we currently use. So why should someone choose the NF-core protein family software to generate families? And for example, why not just use a jackhammer? But some obvious reasons is that it's a suite of different range of operations. Protein families allow for intra-family sequence redundancy removal as well as inter-family redundancy removal. It allows alignment trimming, and it also has an update mechanism. But what about alignment quality? Can we match the quality of manually curated protein families that actual scientists go and curate? We have set up an experiment to do in order to figure that out. First, we're going to collect some sequences from interpro families. And in these sequences, we're going to add some decoy sequences without hits from UniProt. And then we're going to run the NF-core protein families pipeline. And also, after it's done, we're going to run jackhammer on the consensus sequences of the generated families. And finally, we're going to compare and assess the quality of the generated families from both protein families and jackhammer against the original curated families that we've taken from Interpro. So the verdict: if this is successful, we can say that we have an automated way to mimic family curation efforts, but at scale with the protein families pipeline. If not, we will have to revisit the family generation algorithm, perhaps. And well, Nextflow is not allowing module loops at the time, but we have some alternatives that we can create one module that does everything inside in a loop and provide a bit better results. I do not have an answer for you right now because I'm currently running the experiment, so you will have to wait for it, I'm afraid. And what else we're working on right now for the upcoming release, version 1.1.0? First, because I've run the pipeline with different data multiple times that people have given me, I've consistently been finding duplicates in the original input datasets. So we will need to create a quality check pre-processing module that removes these duplicates with 100 identity clustering run. And also maybe some proteins sometimes have duplicate names, even though they have different sequences, which is again very annoying and produces MMSX clustering errors. So we will have to do that as well. Then we will need to create an NF-core module for Trimal to provide it as an alternate mode to ClipKit. They're both software that clip gaps from multiple sequence alignment files. Then we will also input another quality check after we build the families, aiming to discard families that are either very, very small in length or very, very large. This, of course, will be tunable by the end user. Then, yes, as I already told you, we're going to also put in an alternate mode to HMM search with the nail software, and then also filter the results with PyHammer and PyFastX to bring down the job durations. And add another in the inter-family redundancy removal step, because sometimes HMM search might remove superfamilies or smaller families, depending on the size. If the user does not use a very strict identity threshold, just to make sure we have in mind to use a better redundancy filter, comparing two families with a Jacquard similarity index. So we decide we're certain that we're removing a family that's not a subpart of another family. And finally, we will try to skip this strict clustering 4-module sub-workflow with just by using Trimal with the minus clusters option, which just gives the n number that we state of best sequence representatives. Of course, we will try to replace Python sec.io library with PyFastX throughout the modules and benchmark the time that we can save. Also, we need to add NF tests for the remaining local modules and local sub workflows, as well as for the whole pipeline. And finally, we aim to create a new workflow that will allow, if the user inserts HMMs and MSAs from previous executions, they will be able to merge families in the future. So to wrap it up, there's a nice ecosystem of NF core pipelines that are used in metagenomics. I think there are 14 right now. And protein families falls really nice here because any pipelines that generate FASTA protein outputs like MAG and MetaT de novo, they produce these protein FASTA files, and these can be fed directly into protein families, which is very convenient. These are usually millions of proteins. And downstream, after protein families produces protein representatives of these families, this can be fed into protein folds to see the structures and also annotate annotate the proteins based on the 3D structure, and also in the upcoming protein annotator pipeline. I'd like to thank my team, Martin, who really helped with next flow coding for the pipeline. My two bosses, let's say now, Rob Finn from EBI and my previous group leader, George Pavlopoulos, who is also hosting me here. And of course, NF Core community, who really helped with this. Any questions?
So thank you very much. Now we see everyone. Are there any questions from the audience? So otherwise, I might ask a question. I'm a nucleic acid person, so proteins is not really my thing. I was just wondering what is more difficult, to find families for proteins or to find related genes and nucleic acid sequences? Or is it the same, basically?
Because I'm a proteins person and have not played with nucleic acids this much, I'm not sure. I would say, with a wild guess, don't take my word for it, I would say it's easier for proteins to make relationships because there are way more amino acids than nucleic acids, right? So it seems easier to be able to make sure that something falls in the same family. At least that's how I think about it.
Yeah, I see that. I mean, for proteins, you can basically have sequences that are not at all like by sequence related, but they are by function related or by structure. But
Yeah,
pipeline actually figured these out because I don't really think they are comparing structure here, right?
no, we don't play with structures in protein families. I don't want to take work from protein fold. Right? Everybody with his pipeline. But what I can say is that if you want families that are strictly connected like ortholog groups, you can set your identity thresholds to be very high, like 90%, above 80% similarity. But if you want broader families that might have similar sequences, that are evolutionary similar sequences, but with multiple different functions inside the same family, then you can lower your identity thresholds, go down as much as 30%, maybe less for some families. That's how you expand the family with many domains instead of one function, let's say.
Yeah. Thank you. Are there any more questions? So anyone can unmute themselves if they have a question.
Well, you can always ask in the protein families Slack channel.
Thank you. So it seems everyone is at the moment happy. So thank you so much for this talk. I'm always happy to see more non-nucleic acid pipelines in NF Core. So thank you so much. And thanks for the audience for listening in. And I hope to see you for the next Bite Size talk. Bye-bye.
Bye.